So chapter 7, we're going to take a look at what are all of the various things that microbes need to survive. So it kind of lays it out so at the beginning what it goes over in the chapter. So what are certain types of nutrients that different organisms need? Where's their energy source? They all need energy. What kind of transport is required to get things in and out of cells? What are different types of habitats, environments that they live in and thrive in? What are some different types of relationships? And then how, once they've got everything that makes them happy, how is it that they're growing and how do we measure growth? So it's really just looking at what do they need to survive, all the unique things they need to survive, where they survive, relationships they need, and then measuring growth. So we're going to start by looking at what are some of the things that they need. What are some of these nutrients that they need? I'll make it larger. So anytime we're talking about microbial nutrition, they need lots of the same things that we do. They need various elements, they need organic compounds, they need sunlight or some type of energy source, all the different things that we need. Where they get it from could be a little different than from us, but they need very similar things. So just looking at some of the basic requirements they need carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, all of these are all the same types of elements we need for survival. They also need what are considered essential nutrients. These are just substances that they have to get from some source outside of its own cells, which means they can't make any of these particular elements. They have to get them from somewhere else. Now, one group of nutrients is considered organic nutrients. Now, just a little review of what organic is, because you've probably heard the term in other classes as well. Organic just means carbon-based, and usually when it's carbon-based, it's also going to have hydrogen molecules along with it. Example, methane, CH4. It's got that carbon molecule. It also has some hydrogens. Carbohydrate, if you remember the formula for basic carbohydrates, it's always 1 carbon to 2 hydrogen to 1 oxygen. So it's got those carbons and hydrogens. So a lot of organisms need organic nutrients. But there are also inorganic, non-carbon-based nutrients as well that are required. Now, just like us, prokaryotic cells, bacteria, are mostly made up of water. So they're going to have some water regulation. But we're going to talk about first what are the 6 top elements they need. Now, these are the same top 6 elements that we need in our cells as well for survival. Now, in your book, there is a nice big table that goes over a lot of these elements, which is going to be a lot of these next slides. I'm going to kind of skip around until we hit the top slides where it talks about the carbon, the hydrogen, the oxygen, phosphorus, sulfur, and nitrogen. So the first one, so it's table 7.1, so it's the next 17 slides, but I'm going to skip around a little bit. The first is the carbon. It is out of the top 6 here, and I'm like, carbon is the number 1 element that's required. I'm like, we are organic molecules. Bacteria are organic molecules. We're all carbon-based. Now, carbon itself is found out in the environment. It can be found in different types of gases. It can be found in different types of organisms and other living things. And I'm like, but there's a lot of carbon out there that can be available. The next on our list is nitrogen, is the next element. Again, there's lots of nitrogen out there. A lot of nitrogen in the air. What you're breathing in mostly right now is nitrogen, not oxygen. It's just we don't use it for anything. So I'm like, there's lots of nitrogen out there. Organisms, bacteria need it. Oxygen, and we're going to talk a little bit about the oxygen as well, because different bacteria, different organisms require different amounts of oxygen, and some require 0 oxygen whatsoever. They cannot be in an environment where oxygen is present. Now, oxygen is mostly in air, and I'm like, but we can find a little bit in soil as well. Hydrogen, again, a lot of hydrogen is in air in a gas form, but it can also be bound up in various types of elements, lots of organic compounds, and it's found in water. And phosphorus, I was going to say, it's normally bound up in a compound called phosphate, usually in soil. And then sulfur. It's also usually bound up, it's usually found in type of soil sources, but these are all top elements that organisms need. Now, some of them are more necessary than others, the carbon, the oxygen, the hydrogen, and even the nitrogen. So we're going to talk a little bit about some of those. I'm not going to go through all the other little ones. Now, the carbon-based part, because yeah, they require, and we require carbon. We require organic molecules. And so we group organisms based on where do they get their carbon. If they obtain carbon from some organic form, meaning they eat it, they eat organic carbon-based molecules, we consider them heterotrophs. If an organism uses carbon, which is considered an inorganic gas, even though it does have carbon, as its carbon source, we consider it an autotroph. That's usually for organisms that can do photosynthesis. They can take carbon dioxide from the air and turn it into a usable carbon form. Plants can do that. We can't. But there are some bacteria that can do photosynthesis that are autotrophic. So heterotrophs, they get all of their carbon from organic molecules. Heterotrophs, they get all of their carbon from carbon dioxide in the air. Now, for organic molecules, there are some organic compounds that we can't make at all. And I say we, as I guess bacteria, we can't make them either. Bacteria can't make them whatsoever. And so they have to be provided somehow in their nutrients, and somehow in their diet, and they're called growth factors. There are certain amino acids that cells can't make. There are certain vitamins that cells can't make. It's just like we would take a daily vitamin. It's because there's various substances in there that we can't make, and we might not get elsewhere from various diet sources. And so just bacteria, same thing. They can't make certain things in their cells, so they're going to have to get it from somewhere else. It's just called a growth factor. It has to be provided somewhere else. Now, we group organisms based on where they get their carbon. Again, heterotrophs, they get it from other organisms. They can eat other organic carbon-based molecules, just even eating living plants. Autotrophs use carbon dioxide as their carbon source, but we do also like to group organisms not just based on their carbon source, but where do they get all of their energy? Because organisms need energy to be able to build things up, break things down. And so where would they get their energy? We group them into chemotrophs or phototrophs. A chemotroph gains all of its energy from chemical compounds. It means they break down some type of, usually an organic molecule. And anytime you break something down, when you break a bond, you're going to release energy. So we call it a chemotroph. So they're getting all of their energy from breaking down chemical bonds in some type of chemical compound. A phototroph gets all of its energy through photosynthesis. So they're getting their energy from light. Now, we then smush these words together, and we can describe an organism based on where it gets its carbon and where it gets its energy. Example, we can smush photo to autotroph, and we can say, it gets its carbon from carbon dioxide, and it gets its energy from light. A chemoautotroph gets its carbon from carbon dioxide, and it gets its energy from chemical breakdown. Chemoheterotrophs gets its energy from its carbon from organic compounds, and it gets its energy from breaking down chemical compounds. So we can group organisms based on those two things, their energy source and their carbon source. Now, most of the organisms that affect us aren't doing photosynthesis. Most of the organisms that affect us are chemoheterotrophs, where they get their energy from feeding off organic molecules like us, and they get their energy from the breakdown of organic chemicals. So this is just showing the photoautotrophs. A lot of them, these are the ones that do photosynthesis. Again, these are generally not the ones that are going to cause us harm. They're out there in the environment, though. There are a lot of bacteria. There are a lot of organisms out there. Generally, we don't care about the ones that don't affect us personally, but they can do other things in the environment that, again, help makes the world go round. And I'm like, we in this class are going to focus mostly on the ones that are going to affect us as humans. So we don't talk a lot about the autotrophs. We again talk mostly about the chemoheterotrophs that get their energy and their carbon source from organic compounds and breaking them down. And some of these are going to be saprobes that feed off dead organisms, fungus, we talk about fungus, and a lot of them are going to be parasites. Most of the ones we talk about are parasites, which means they're going to get their nutrients from us, causing us harm. We call them pathogens. It means they are going to cause us harm as they feed off of us. Now, other than different types of elements that organisms need, they also need to regulate what goes in and out of their cells. And so there's a couple different transport mechanisms. This should be reviewed from any previous A&P classes or biology classes you may have had, so I'm not going to spend a ton of time on these slides. Just a quick review on different transports moving things in and out of cells. Passive transport doesn't require any energy whatsoever. Examples: diffusion, osmosis, facilitated diffusion. Active transport does require energy in the form of ATP. Usually requires some type of carrier protein to move things from one side of the cell to another side of the cell. This is just showing diffusion, going from a higher concentration to a lower concentration. Osmosis, it's the moving of water from a high concentration of water to a lower concentration of water. Water wants to go where there's not as much water. So this is just showing where water is moving. And I'm like, in these 3 slides. My little note, it does have a little question on the bottom though. Even as the solution becomes diluted, there will still be osmosis into the sac, meaning water's still going to want to go into that little sac there. It says equilibrium will not occur because the solutions can never become equal. Why? Well, the main reason why is everything is always moving. Every chemical, every molecule is always in motion. And so as one thing leaves the sac, one thing moves in one direction or the other, everything then becomes unbalanced. So as stuff is always moving, it's always becoming unbalanced and then trying to balance itself back out. Now, when we talk about moving of water, there are a couple different terms based on the direction of water movement. An isotonic solution, the iso means the same. When you talk about an isosceles triangle, has similar same sides. An isotonic solution means the water concentration is the same inside and out, which means if water goes into the cell, the same amount's going to go outside of the cell. A hypotonic solution means that there is lots of water outside of the cell, and it has a very low solute concentration, so water is going to want to move into a cell. Now, depending on how much water can move into the cell, it could cause the cell to burst. Luckily, bacteria cells do have cell walls that can try to prevent the cell from bursting, but it may not always. But at least it can slow it. But cells are going to swell up. It's not great for a cell. A hypertonic solution does the opposite. Water is going to want to leave the cell, and the cell can shrivel up. Now, there are some cells that don't even have walls. Most do, but there are some cells that don't have cell walls. So an isotonic solution, same things. Things are going to move in and out of the cell. In a hypotonic solution, if there's no wall, it's a guaranteed that cell's going to burst. That's what happens to our cells. And in a hypertonic solution, they definitely are going to shrink up to almost nothing. So cell walls in a bacteria do help it to withstand water differences in different solutions. Facilitated transport or diffusion, it's still diffusion. It's still going high to low concentration. It just requires some type of carrier protein to move things through. Group translocation, it's going to use some type of carrier protein, and it's going to move multiple groups of things, so multiple things across. Instead of just these little ovals and little squares, it's going to move them both together across. Now, when we bring things in, it's not just moving water inside of cell. We can bring other things inside of cells as well. Endocytosis just means it brings it in. Phagocytosis, phago means eat, It's when cells eat things. They ingest large substances. They may even ingest other cells. An example of a pseudopod, or not, an example of phagocytosis, our white blood cells do phagocytosis all the time. They eat bacteria. They go around, form what's considered a pseudopod or a little false foot around the bacteria, bring it in, break it down, and then they keep doing it. Penocytosis is bringing fluids or very small dissolved substances inside of a cell. This is just showing that endocytosis, how it's going to go around various substances and bring it in. That's the phagocytosis. Now, other than various elements, and other than they have to regulate what goes in and out of the cell, and I'm like, where an organism lives and what kind of environment it likes is going to be different from each other. Now, the environment, the type of environment they prefer is considered its niche. Now, some factors that affect an organism's niche, where it likes to live, what are different factors that it requires. It may require a specific type of temperature. It may require oxygen, certain levels of oxygen, a certain pH, some type of osmotic pressure or a barometric pressure. I'm like, these are all different environments. I'm like, barometric pressure, living on the top of a mountain is going to be completely different than living at the bottom of an ocean. And I'm like, but different organisms have developed their own environment, their own niche for that particular habitat. So we're going to talk about some of these factors that are going to be unique to certain organisms. And the first one is temperature. And I'm like, organisms have optimal temperature growth. It's the temperature that they grow best at. They're going to grow the fastest. They're going to be the happiest. But it doesn't mean it's the only temperature they can grow at. There is a minimal temperature. It can get real cold up into a certain point that they're still going to grow slowly. But they can still grow. Same thing with maximum. And I'm like, they can still grow at higher temperatures above their optimal temperature. They're just not as happy, so it's not going to be an optimal growth. Now, a couple different organisms, or a couple different temperature groups that different organisms like to live in. And I'm like, this is showing the different temperatures in what we call as psychrophiles, psychrotrophs, mesophiles, thermophiles, and extreme thermophiles. Now, out of just looking at these three, the group of organisms that affect us are going to be the mesophiles. Now, I should note that I like to break apart words. The phil of mesophile means likes. So mesophiles means it likes moderate. Meso means moderate or middle temperatures. And so it likes temperatures around 20 to 40 degrees Celsius. And so on here, it's anything in the green. Its optimal temperature, if you notice, is around 37 degrees Celsius. Body temperature. These things are going to grow best once they get in their body. But that doesn't mean they can't grow outside of our body. They have still a pretty wide range of temperatures that they can grow in. But they grow best once they get inside of our body. Now, if they're colder than that, and I'm like, we consider them psychrotrophs, or if they grow in only cold temperatures, psychrophiles, which my way for remembering it is, you might be a little psychro if you like living in the cold temperatures. I know we live in Wisconsin, and I know it can get cold here in the winter for 6 months of the year. But these organisms thrive. They only grow if it is freezing cold temperature. If you tried to take one of these organisms and put it in us, it wouldn't even grow. I mean, there's almost zero overlap. So I'm like, it would be boiling hot if you put one of these organisms in us. Now, the other extreme are thermophiles. Therm means warmth, and so these like warm temperatures. They like temperatures greater than 45 degrees Celsius. If you tried to put one of these organisms inside of us, it would freeze. It wouldn't grow at all. These are organisms that grow and thrive in hot springs. And so I'm like, they require that hot boiling temperature. Now again, and I'm like, they don't affect us, so we generally don't care about them all that much. But there are lots of organisms out there. They just have their own different temperature requirements. Another requirement for different environments is whether there is oxygen present or not. Now, oxygen is used in a lot of different chemical processes, and unfortunately, sometimes oxygen can turn into toxic forms of oxygen when it becomes unstable. Now, most cells have developed enzymes called superoxide dismutase, which is long, and I'm like, it's an enzyme, it ends in ace. Most enzymes end in ace. It's an enzyme that can kind of neutralize the superoxide. That's where they get the superoxide ion. Catalase, same thing. It can neutralize some of these toxic forms of oxygen, which means they can thrive in an aerobic, oxygen-containing environment. So as they use oxygen and they make toxic forms of oxygen, they can neutralize it. Now, if you're an organism and you don't make catalase, or you don't make superoxide dismutase, and you have used some oxygen that was around and you made a toxic form of oxygen, it would then become deadly. And so that's where we get into the anaerobic organisms. They can't neutralize those toxic forms of oxygen. Now, if something requires oxygen in their environment, they need it in various chemical processes, we call them obligate aerobes. I always like to think it's just obligate means it's required. They cannot grow without it. A facultative anaerobe says it uses oxygen, but it can grow without it. So my kind of way to remember is facultative. I always think, it's like faculty, teachers. We can do it all. Like, we could use oxygen if it's there, but we can also, survive if it's not there. It's not required. They usually do a little better and grow better if oxygen's present, but if oxygen is not present, it won't kill them. And then there's a group what's called microaerophilic. Again, I like to break apart words. The "phil" part means it likes, micro means small, and air refers to oxygen. It just means it requires a small amount of oxygen. It needs oxygen, but it prefers it in a smaller quantity. It doesn't like as much oxygen. We're breathing in around 20% oxygen right now in our air. It would prefer a smaller number to be happier. Now, the organisms that cannot use oxygen are called anaerobes. Now, an obligate anaerobe, again, obligate means it's required. It is required that they do not have oxygen. They cannot survive in an aerobic environment. And then we do have organisms that are called aerotolerant anaerobes. It means they don't use oxygen, but they can still survive and grow if it's around. So they're anaerobes because they don't use oxygen whatsoever. But if oxygen present, it won't kill them. So I'm like, they're just tolerant of it. It doesn't kill them. They just don't use it. They're just like, whatever. It's there. I just don't need it. So they're still anaerobes, but they're just oxygen tolerant. Now, we can actually determine different organisms are specific for their oxygen requirement in labs, and we don't know what a culture, we don't know what kind of specimen a patient may have. We can take a bacterial sample, we can put it in different types of media, and we can look to see where does it grow in a tube. So we can narrow it down based on its oxygen requirement. In a tube, and I'm like, at the very bottom of a tube, there's no oxygen down there. The oxygen can't get down there. And so if organisms are growing down in the bottom where there's no oxygen, anaerobic. If they're growing this one, like there's nothing growing at the top. If organisms are only going to grow at the top where there is oxygen present, well, these are aerobic organisms. If they're going to grow throughout, these are usually your aerotolerant. It's like they're not using oxygen. There's no preference for oxygen. And I'm like, and so we can narrow different organisms down in the lab based on their oxygen requirement. Now, if we're going to, again, we don't always know organisms, what is their oxygen requirement. And it's not the only gas requirement for some of them. Carbon dioxide is present as well. And so we have to be concerned about what kind of environment, oxygen-wise, we give our organisms in a lab. But for some organisms, we also have to be aware of various carbon dioxide environments as well. Most microbes require some CO2 somewhere in their metabolism, and that CO2 is enough in the air to be around. However, there are some organisms that are what we call capnophilic or capnophiles. Phile means it likes, capno, not sure where it comes from, means carbon dioxide. And they grow best if there's higher amounts of carbon dioxide. So in a lab, we would have to put them in an environment where there's higher amounts of carbon dioxide. Another factor that can affect microbes' growth is their pH. Most organisms that affect us are neutrophils. They like a neutral pH between 6 and 8. However, there are different environments, pH-wise, even in the human body, and different organisms require different pHs. Those that like a more acidic pH are considered acidophiles. Ones that like extreme alkaline or basic pHs are alkalinophiles. Your stomach has a low pH, and there are bacteria that can grow and thrive in your stomach. They would be acidophiles. And I'm like, they've developed ways to survive that acid environment. Another environment are the hypotonic or isotonic conditions. We're looking at what is the water requirement. And I'm like, do they require 100% water type of environment? Do they require a specific percentage of solute in there? So some, what we call as osmophiles, they require a higher concentration of salt. These are the ones that grow best in a salty environment. They can grow in the Great Salt Lake. What they thought, there's a reason why they thought it was called the Dead Sea. It was so salty that nothing could survive in the Dead Sea. Well, there's bacteria that are living in there, so it's not dead. And they can grow in that really high concentration of salt, where there's that really low concentration of water. There are certain bacteria that require that specific osmotic percentage. There are some organisms, they don't require a high concentration of solute like salt, but they tolerate it. An example on you guys is a bacteria, anything in the genus Staphylococcus. They can survive a high salt concentration. These are most of the bacteria, the staph bacteria, that like to live on your skin. Your skin is salty, especially when you sweat a lot. And that salt inhibits most bacteria. Most bacteria don't like salt. But Staphylococcus are tolerant of it. They don't use it, but they're tolerant of it, and it won't kill them when you get salty. Another environmental factor is pressure. There are some organisms that survive under extreme pressure. These would be the organisms that live in places like the bottom of the ocean, those deep trenches, where that pressure is so high that if you brought them to the surface, they would explode. They need that pressure to keep them together. Again, organisms that require a high barometric pressure, not going to affect our humans. Now, other than different types of factors in the environment that they require, there are different relationships that organisms have with each other. Some are required relationships, others they just thrive better at different relationships. Now, a symbiotic relationship just means that organisms that live in some type of close relationship with each other. Sometimes it's a required relationship by both organisms, sometimes it's just a required relationship by one. Couple examples of these symbiotic relationships. Mutualism means both members benefit. So you've got two organisms living together, both of them are benefiting from the relationship. Commensalism means one member benefits and one member is not harmed whatsoever. They're not harmed, they're not helped, they're just there. An example would be some of those staph bacteria on your skin. They benefit because they have a home. You gave them a home. They're benefiting. But you're not harmed. You have bacteria all over your skin, and they're with you. No matter how many times you shower and wash your hand, you still have bacteria on your skin. They're not causing you any harm. They're your normal flora bacteria. And I'm like, but they are benefiting from that commensal relationship because they now have a home. Example of a mutualistic relationship with bacteria is you have bacteria in your digestive tract. And some bacteria do help break food down. They help in digestion. Some help make various vitamins or precursor vitamins. And so they are helpful to you. We benefit from them. And they benefit because you provide them a home and feed them. Parasitism are the ones you got to be worried about, as it means one organism benefits at the expense of the other one. And that's going to be any type of pathogen. Anything that causes us harm to its benefit is considered a parasite. Now, some of these mutualistic relationships, if they are required, where both organisms have to be together, we call them obligate mutualistic relationships. If they could be separated and live apart from each other, usually they do better if they're together, but if they could live apart from each other, we just call them non-obligate mutualism. Now, on here, at the top it says 1 out of 21. These are the next 21 slides. These next 21 slides all are really taking a look at figure 713 in your book. It has a lot of these different types of relationship and lots of different examples of different types of mutualistic relationships, parasites, commensalism, specific obligate mutualism, and lots of pictures. So if you want to take a look at those in your book, figure 713. Some of these you may have never realized how many organisms you have living on you, and it gives some examples of some. But it gives some good examples with lots of pictures. But I'm going to scroll ahead to what we call biofilms. Now, the word biofilms, it's a symbiotic relationship. So these are organisms living together. Bio itself means living, film means covering. So it's considered a living covering. These are organisms that are living together and covering the surface of something. Now, they usually use some type of chemicals called quorum sensing, some type of protein that allow them to move towards some particular surface. It's like, oh, there's something good over there. We should all go over there. And then we're going to stick together. They make a nice sticky matrix to cover themselves. They all stick to each other. They send out more chemicals to attract others. And they eventually form a full covering on a surface. Now, we have biofilms that cover various surfaces of us. Now, generally, when bacteria hang out together, this is showing all bacteria of one species, but they don't have to be the same species. When you have lots of bacteria hanging out together, for them, it's strength in numbers. That is, there is some protection there when they're all together. That sticky matrix allows them to stick and help them find a home. It allows them to stick and not get washed off easily. And so I'm like, it's a little strength in numbers for them. It's usually good. Now, in general, anything that's good for bacteria is bad for us. An example of a biofilm, very common biofilm found on you is, we've got biofilms anywhere on our mucous membranes, but in your mouth, specifically on your teeth. If you've ever gotten to the end of a day and your teeth feel kind of a little furry, you could scrape them off, and you could even scrape some stuff off your teeth. That's biofilm right there. That is bacteria and that sticky matrix that's holding bacteria on your teeth. Now, I'm kind of bummed because if we would be in lab, one of the labs we've done before in the past is we've scraped our teeth and then we've stained the bacteria so you can see how many bacteria are all over your teeth. It doesn't matter if you just brush your teeth. You always have bacteria in your teeth. They are sticking to your teeth. Now, again, it's good for them. They have a home. Bad for us because that matrix can harden into what we call as tartar. That's where if you don't brush very well and you go to the dentist, that hygienist has to chisel that tartar off. But as bacteria are hanging out in your teeth, they can do fermentation of sugar that you eat. So when you eat sugar, they can break that sugar down, and anytime you ferment a sugar, you always get an acid. And that acid breaks down the enamel on your teeth. So when they talk about, oh, sugar causes cavities, well, yes, but it's the bacteria that eat the sugar to make the acid to then cause cavities. So yes, sugar causes cavities, but not directly. It's the bacteria that eat the sugar that makes the acid that causes the cavities. Now, this is just a little description of this particular picture. So yes, we are a rich environment for lots of different organisms. You have organisms all over you, inside and out. You are not alone. Now, when organisms are happy, they've got their perfect environment. They have all the nutrients, all the energy, any possible relationships that they possibly need. When they are happy, they are going to grow. And bacteria grow through a process called binary fission. The bi part means two, fission means split. It's the opposite of fusion. So they are going to grow and then split into two, binary fission. So they just enlarge, they double everything, and they divide into two. Now, I was going to say, this is showing, kind of describing this picture. So they are going to grow, they're going to double their genetic information, and then they're going to split. They don't do anything, cool like mitosis or meiosis. They just grow, double, and split, and that's it. Now, because by the end of binary fission, we start with one cell, and now we have two. Well, those two cells are going to do exactly what the original one did. They are going to grow and then divide. And so we get what's considered a doubling. And so they are constantly going to be doubling. Now, the time that it takes for them to grow and divide is called their doubling time, also called their generation time. Now, generation times or doubling times can vary from minutes to days. A lot of bacteria have a doubling time of 30 minutes or less if they're in their optimal temperature, oxygen requirement, and everything else you've given them. Now, this is just showing, and I'm like, that we start with one. After one generation time, it becomes two. Those are going to all divide. After another generation time, they become 4. Those are going to double, it becomes 8. Those are going to double, it becomes 16, and so on. And you can get really rapid, really fast growth of bacteria in a short amount of time. Now, this growth, where it is a doubling, and I'm like that, yeah, we went from 1 to 2, and then, now we only have 4. But now all of a sudden we jump up to 32, and then 64, and 128, and we get this really fast doubling time. This is called logarithmic growth. We don't grow logarithmically. Most organisms do not just keep doubling. I'm like, but bacteria can do this in a very short amount of time, this logarithmic growth. Now, to know if bacteria did grow, there's a couple different ways we can measure and determine if bacteria grew or not. One of them is called a viable plate count. Because sometimes we want to know, how many bacteria are there? Did they grow? Did they not grow? Did we give them enough time if we want a specific number? Well, if we have a flask that has a whole bunch of bacteria in it, we can sit there and but again, we don't know how many bacteria, but we can put samples in tubes, we can grow them up on plates, and we can measure over time. And I'm like, how many bacteria are there after every 60 minutes? And we can start to viable plate counts. And I'm like, to know, what, if we're looking for a specific concentration or we want to know what the doubling time is, we can look to see how fast they're growing. Now, most organisms, bacteria, again, if you make them happy, they are going to have that exponential, that logarithmic growth. But they just don't go straight into logarithmic growth once they're happy. There is this little stage. First, there's a lag phase that as soon as you put bacteria on a plate in a lab or bacteria get into your body, they don't just start doubling. They are kind of making themselves at home. They might be starting to ingest whatever food source is there. They're just starting to get ready to start to divide. So they have what's considered a lag phase. Then, once they've got everything they need, they go into what's known as the exponential growth phase. That's your logarithmic growth, that really fast doubling. Then after a while, they're going to start to have less food available, and they've got lots of waste products around. And so they're going to be still growing, but you have the same number growing as you have the same number of organisms dying. And so we have what's known as the stationary phase. And then you have the death phase, where you have more organisms dying than growing. There's still some growing, but you have more dying than are growing. Now, generally in a lab, we always want to try to look at bacteria when they're in this exponential logarithmic growth phase. Because they are the happiest bacteria. You're not going to see dead ones underneath a microscope because they're not going to stain very well. So we always try to grab organisms when they're in that exponential growth phase. Another way to look to see if organisms have grown, at least an approximation, how much growth has happened, is just to look at turbidity. cloudiness. And I'm like, here's 2 tubes of media. This one's a whole lot cloudier than this one. The more cloudy it is, the more bacteria it is. We don't have an exact count of how many bacteria are in there. And I'm like, but we know there's a lot of bacteria in there. Now, there are some machines that can look at turbidity. And I'm like, because if you shine a light through it, if something's very turbid, light bounces off all the particles in there, and you're not going to have as much light shining through. And I'm like, so it kind of just gives you a number scale for turbidity, but it doesn't tell you how many organisms there are. But the more cloudy it is, the more bacteria there are. Another way to analyze if organisms grew or not is just count them. Is we could put them, there are special slides that we can put these on, and they have little grids on them, and we know that there is a specific volume of fluid in between a cover slip and the glass slide itself. And so underneath the microscope, you could count the bacteria and say, oh, there's 9 bacteria in this tiny little grid, and we know that there's 1 microliter of fluid. God, 9 bacteria per 1 microliter. And we've got a pretty good estimate of how many bacteria are in a solution. Other ways that we can do it, some type of machine. One's called a Coulter counter, one's called flow cytometry. They work very similar to each other. Is if you have bacteria in a solution, you can put them in a machine. The Coulter counter uses electricity, that anytime a bacteria interrupts the electrical flow, it counts the bacteria. So the more bacteria, the more interruptions in electrical flow, and it gives a pretty good estimate. Flow cytometry, similar, except instead of electrical current going through, it uses a laser light, and a laser light, as anytime a bacteria goes in front of the laser, it counts the bacteria. So the more bacteria, the more times that laser light is interrupted. So it gives a pretty good estimate on how many bacteria are in a particular solution. So in this PowerPoint, again, we talked about what are different types of energy sources, different types of elements that organisms were required, what are different types of relationships that organisms need to survive in. We talked about different types of environments, oxygen in the environment, temperatures. We talked about, there's a lot of stuff in this PowerPoint. Talked about moving things in and outside of cells, and we talked about at the end that once they're all happy, how do they divide and how can we measure bacterial growth if it has occurred. So lots of new terms in there, some review things. Again, if you have any questions on the PowerPoint or anything in this recording, please send me an email.